Welcome, science enthusiasts. Today, we delve into the fascinating realm of neuroscience as we explore the groundbreaking research that has unraveled the intricate effects of ketamine on the brain's entire dopaminergic system. Scientists have embarked on a journey to map the diverse and complex interactions that take place within this crucial neural network when exposed to ketamine. Join us on this intellectual adventure as we uncover the latest findings, shedding light on how this powerful substance influences the delicate balance of neurotransmitters in the brain. Get ready for a mind-expanding exploration into the world of ketamine and its impact on the intricate machinery of our brain's dopaminergic system. Researchers have mapped the effects of long-term ketamine exposure on the dopamine pathway in mice's brains. Ketamine is a medication that is frequently used for anesthesia and is now increasingly being used to treat depression. The work was just published in the journal Cell Reports. The findings reveal evidence of significant structural change in the brain's dopaminergic system in response to persistent ketamine administration. Stated differently, the research indicates that prolonged exposure to ketamine might cause significant alterations in the brain regions associated with dopamine, a neurotransmitter that is essential for mood, motivation, and reward systems. So why concentrate on ketamine? Ketamine has been used for more than 50 years as a dissociative anesthetic, but in recent years, its quick-acting antidepressant effects have drawn attention. Its antidepressant effect is transient, though, thus ongoing or prolonged medication is frequently required. Possible adverse effects are a worry with this prolonged use. Ketamine's wide clinical importance and the dangers of long-term use, especially at higher doses, make a thorough understanding of the drug's effects on the brain imperative. Our main goal was to learn more about if and how long-term drug use affects the brain's neuronal networks. The effects of ketamine on the entire brain were the main focus of this investigation, according to research author Raju Tomar, an associate professor of biological science at Columbia University. For more than 50 years, ketamine has been utilized as a dissociative anesthetic and has been applied in a variety of clinical settings, such as a transformative antidepressant. Ketamine does have an antidepressant effect, but it wears off quickly, about a week, so long-term maintenance therapy is required. There is a substantial danger of negative effects from this continual consumption. In order to fully and impartially evaluate the effects of different ketamine dosages on the brain at the subcellular level and to comprehend any possible underlying molecular pathways, our goal was to create whole brain mapping tools. Male mice that were specifically developed for this kind of research were used in the study. The mice, which were 8 to 10 weeks old, were given varied amounts of ketamine or a saline control for 1, 5, and 10 days, respectively. The ketamine doses employed were 30 mg per kilogram of body weight and 100 mg per kilogram, imitating both therapeutic and high-dose recreational usage. By using advanced brain clearing and staining methods, the researchers were able to view and examine every cell in the brain at a high resolution. They employed a state-of-the-art technique called light sheet microscopy, which produces precise three-dimensional images of brain tissue. Processing massive datasets, about 100 terabytes, during data analysis necessitated the creation of specialist software in order to ensure thorough and precise analysis. By focusing on the complete dopaminergic system in mice and evaluating the drug's impacts on neuron populations and their projections, the study hoped to provide a comprehensive understanding of ketamine's various effects on brain function and structure. The brains of mice given lower dosages of ketamine demonstrated an increase in dopamine-related neurons in certain regions of the hypothalamus after 10 days of treatment. This area of the brain is critical for controlling a wide range of essential functions, such as mood, appetite, and sleep. On the other hand, in certain midbrain regions, greater dosages of ketamine resulted with a reduction in the number of neurons. Higher ketamine dosages may have a substantial effect on these brain regions since they are linked to behavioral state regulation. Additionally, the study showed that long-term exposure to ketamine changed the ways in which neurons project themselves to other parts of the brain, altering the patterns of neuronal connections. The prefrontal cortex, which is involved in social behavior and decision-making, and other regions linked to higher-order cognitive activities, showed a notable increase in the density of these projections. On the other hand, areas processing spatial and auditory information showed a reduction in neural projections. According to this research, ketamine may reorganize the brain's communication networks, which could alter how information is assimilated and processed. Co-author Malika Datta said, "The restructuring of the brain's dopamine system that we see after repeated ketamine use may be linked to cognitive behavioral changes over time." The role of untranslated messenger RNA, mRNA, in the brain's ketamine reaction was another important finding. Untranslated mRNA is a type of genetic information that is not used right away by neurons to produce proteins. These untranslated mRNAs, the researchers discovered, contribute to the brain's capacity to adapt to long-term ketamine treatment. To be more precise, they offer a reserve that can be quickly drawn from to adjust the quantity of dopamine-producing neurons in response to the medication. This process suggests that the brain has a multi-layered regulatory layer that enables rapid adaptation to outside inputs, such as drug exposure. The thorough investigation offered a clear picture of the wide-ranging structural changes made to the brain by ketamine. A divergent effect of ketamine, affecting distinct brain regions in contradictory ways, is shown by the decrease in midbrain regions and the increase in neuron counts in specific hypothalamic areas. This discrepancy implies that ketamine has a very complicated and location-specific effect on the brain. Prolonged use of ketamine can profoundly reshape the brain's neural circuitry, Tomer told SciPost. This study also emphasizes the need for targeted delivery methods by demonstrating how different brain regions can respond differently to similar types of cells. These results have important ramifications for our knowledge of how the brain works and how brain illnesses are treated. This work advances our knowledge of the neurological basis of mental health disorders such as schizophrenia and depression by demonstrating the wide-ranging effects of a psychoactive drug like ketamine. It also emphasizes how crucial it is to think about the dosage and extended use of medications like ketamine when utilizing them therapeutically. According to co-author Bradley Miller, a psychiatrist and neuroscientist at Columbia, ketamine rapidly resolves depression in many patients with treatment-resistant depression, and it is being investigated for longer-term use to prevent the relapse of depression. This study reveals how ketamine rewires the brain with repeated use. Developing focused therapies that successfully treat depression without some of the undesirable side effects of ketamine requires taking this crucial step. Although the study demonstrates alterations in dopamine-related brain regions, it's crucial to keep in mind that same areas also house neurons that are involved in the production of other neurotransmitters. Therefore, dopamine may not be the only factor contributing to the modifications that have been seen. According to Tomer, the effect of ketamine on the brain's dopamine systems was specifically investigated in this study. Future studies are required to investigate its impact on the brain's other systems. Furthermore, more thorough research is required to determine the precise neuronal and molecular pathways that permit structural remodeling and the related behavioral modifications. Thank you for joining us on this enlightening journey through the labyrinth of the brain's dopaminergic system under the influence of ketamine. The world of neuroscience is constantly evolving, and today, we've touched upon groundbreaking research that provides a deeper understanding of how this powerful substance interacts with our brain's intricate network. If you found this exploration intriguing, don't forget to hit the like button, share with fellow science enthusiasts, and subscribe for more captivating insights into the realms of science and discovery. Leave your thoughts and questions in the comments below. We love hearing from our curious community. Stay curious, stay informed, and until next time, keep exploring the wonders of the mind with us. Remember, the pursuit of knowledge knows no bounds. See you in the next video.